I'd like to tell you a little bit about our presentation today. I spoke about two aspects of nutrient stress and inflammation and the link between nutrients and body inflammation. The first was on a process called insulin resistance. This is the root cause of diabetes. Insulin resistance is a major problem that we do not understand. And my presentation described a novel molecule that when injected into the body, either under the skin or in the bloodstream, called FGF1, an old molecule but with a new function. And even though it is more than 30 years old, it has never been known to have a role in metabolism. And as our recent work has shown, it's a potent metabolic regulator, and if given to mice even a single time, it's able to lower glucose to very normal or physiologic levels, even in highly diabetic mice. And this leads us to be hopeful that it might be used as a means to control glucose and insulin resistance in humans. So that was the first part of our study. The second part, I described a small molecule, chemical, that mimics bile acids. This is a digestive enzyme, a digestive substance that's released into the intestine when you eat. And everyone knows about bile acids, but we think these are actually hormones, bile acid hormones. They interact with bile acid receptors, and we made a chemical that mimics the hormonal response. And as a result, the chemical is like an imaginary meal in a pill. That is, that you take the chemical and the body thinks that you've had a meal, triggers the bile acid receptor in your intestine, and as a result, you mobilize many different sites in the body. The drug does not get in, but the intestinal response triggers many internal responses, and as a result, high-fat fed animals begin to lose weight. They begin to normalize insulin, and they have less inflammation. So we're also very excited about a drug that does not get in the body that has big effects. And so that was the nature of my presentation for this talk.